Sds-Page is a technique used for the separation of protein based on their molecular weight. It is commonly used to obtain high-resolution separation of complex mixture of protein. The method initially denatured the protein that will undergo electrophoresis. It is a technique widely used in forensic, genetic, biotechnology, and molecular biology to separate the protein molecule based on their electrophoretic mobility. The principle of Sds-Page states that the charged molecule migrate to electron with the opposite sign when placed in an electric field. The separation of the charged molecule depends to the relative mobility of charged species. The smaller molecule migrate faster due to less resistance during electrophoresis. The structure and the charge of protein also influence the rate of migration. Sodium dodecyl sulfate and polyacrylamide eliminate the influence of structure and charge of protein, and the protein are separate based on the length of polypeptide chain.
First, insert a sharp plate and 1 mm spacer plate into the casting frame. Make sure the feet of the casting frame are on the bottom and level together. Then, tighten the clamp to secure the plate's position. Next, install the casting frame with the plates into the casting stand and ensure the setup is tight and secured. Make sure there is no leak occur by seal it with the sealing tape. Check for the seal by pipetting DD water halfway up to the plates. Observe for any leak occurrence. If there is no leaks occur, remove DD water by using clean wipes or filter paper until it is almost dry. When the gel casting stand is set up, prepare 12% resolving gel solution in 50 ml Falcon tube. Add 3.3 ml of DD water, 4 ml of 30% acrylamide solution, 2.5 ml of lower stock solution, and 100 microliter of 10% SDS together. Please note that liquid acrylamide is neurotoxic and should be handled with caution. Right before we pour the gel, add 10 microliter of 10% APS and 4 microliter of TMAT. Due to its strong odor, TMAT must be added under fume hood. TMAT catalyzes polymerization of acrylamide. If TMAT is added too early, the gel will set right before it is ready to be pipetted in between the plates. Mix it gently and immediately pipette the resolving gel solution in between the plates by using glass pipettes until it reaches just above the green line or approximately 1 cm from the top of the sharp plate. Always monitor to make sure there is no leak occur. Pipette 200 microliter of isopropanol onto the resolving gel. A thin layer will be formed. While waiting the resolving gel to be polymerized, prepare stacking gel solution in another 50 ml Falcon tube. Mix 1.35 ml DD water, 0.335 ml of acrylamide solution, 0.25 ml of upper stock solution, and 20 microliter of 10% SDS together. Roughly after 30 minutes, resolving gel form. Proceed with draining the isopropanol by rinse the top of the gel with DD water and dry it with clean wipes. Right before we pour the stacking gel, add 20 microliter of 10% APS and 2 microliter of TMAT into the stacking gel solution and mix it well. Immediately, pipette about 1 ml of stacking gel solution on top of the polymerized resolving gel. Fill it to the top and insert the comb to form the well. Wait for the gel to be polymerized. Once the gel is formed, gently remove the comb and unclip the casting frame with the gel from the casting stand. Then, carefully remove the glass plate from the casting frame. The plate is then wrapped in the wet paper towel and stored in the cold room until it is ready to be used.
To prepare samples for Sds-Page, label the microfiche tubes corresponding to the samples. Pipette 40 microliters of the fish extract into the microfiche tube and add 40 microliters of sample buffer. Heat the tube at 100 degrees Celsius for 3 minutes, then pulse centrifuge for 5 seconds. Remove the gel from the saran wrap and paper towel. Tightly secure the gel with the short plate facing inwards onto the running module. Each running module can hold 2 gels. If only one gel is being run, use the buffer them in place of the second gel. Each tank can accommodate 2 running modules, which will be 4 gels altogether. Insert the running module into the tank. Make sure the module properly inserts into the slot in the tank. At this point, check to see if the lid fits appropriately. Pour the 10 times diluted running buffer solution into the running module between the 2 plates until wells on the top plates are fully submerged. Check to make sure the running modules aren't leaking any buffer solution into the tank. The tank only needs to be filled halfway with the buffer solution if running 2 gels. The samples are ready to load. Remove the comb to see each well clearly. Install the loading guide on the gel to make it easier to load the samples properly each lane. Carefully load 10 microliters of sample solution in each well. The pipette tip should be inserted into the well in between plates deep enough to prevent overflow of the sample to the neighboring lanes. Slowly release the sample solution into the lane. When complete, remove the pipette tip, change the tip, then load another sample. For the molecular weight marker, load 10 microliters into the well and it should be loaded in one of the center lanes for easy comparison.
Load the samples and close the lid on the tank after all samples are loaded. Insert the electrode properly into the power supply. Make sure red wire is inserted into red slot and black wire into black slot. Set the power to 20 mA per plate and run for 1 hour. Initially observe the sample or standard solution to be condensed into a thin band through the staking gel. The band will gradually migrate down the resolving gel. Stop the run when the blue dyes have reached close to the bottom of the gel. Unplug the power supply, then remove the running module from the tank and drain the running buffer. Remove the glass plates carefully from the running module and carefully use the gel from the glass plates. Trim off the staking gel carefully before proceeding to staining. Transfer the gel into a square petri dish. Pour the Comasi blue staining solution into the petri dish, submerging the gel fully. Place the square petri dish in a metal tray to avoid spillage in the incubator, then place in a 50°C incubator to stain for 30 minutes. Remove the gel with a tweezer and make sure clear bands on the gels can be observed before discarding the staining solution and replacing it with the destaining solution. When destaining, add a piece of Kim wipe or absorbent paper towel to help absorb the dye. Destain at 50°C for 30 minutes with periodic swirling and change the destaining solution for every 15 minutes until the background of the gel is transparent. Store the gel in DD water when the destaining is complete.
Western blot is a technique commonly employed in the field of molecular biology to identify and analyze specific proteins within a given sample. And in this method, the proteins in the sample are initially denatured by subjecting them to high temperatures in a boiling water bath for a short period of time. After being denatured, the proteins are separated using Sds-Page. Once the separation is finished, the proteins in the gel are moved to a specific membrane called PVDF membrane, or also known as polyvinylidene fluoride membrane. And these are two methods by which this transfer is accomplished. One method involves placing the membrane directly on the gel and stacking filter papers on top, adding some weight to secure it. And then the entire set is placed in a buffer solution. As the buffer solution ascends through capillary action, the proteins follow suit and ultimately adhere to the membrane. Another method used to perform this transfer is electroblotting. For this technique, the PVDF membrane is carefully positioned on top of the gel and secured between filter papers. This entire set is then moved to an electrophoresis tank. When an electric field is applied, the protein is drawn toward the positively charged electrode and is then transferred onto the membrane. And now we need to identify the target protein by utilizing an antibody. On the other hand, a membrane can also be bound by an antibody. In order to prevent this non-specific binding, it is necessary to block the membrane. And this process involves treating the membrane with a solution containing dilute PSA or milk protein as well as gentle detergent. And this protein has the ability to bind to various parts of the membrane, excluding the specific region where the protein bands are located. And this process is referred to as membrane blocking. After the membrane is obstructed, it undergoes treatment with an antibody that is selectively attached to the protein of interest. This is referred to as the primary antibody. For the detection of primary antibody binding, a secondary antibody will be utilized, and the secondary antibody is connected to a reporter enzyme that transforms a substrate into a visible product, making it simple to identify.